Hi, welcome back to the Jerusalem Science Contest. We're now in the 5th week, which puts us at the halfway mark. And this week we're going to be looking at chapters 13, Plant and Animal Diseases, and chapter 14, Weed Science. And I've chosen for my talk today a topic that is mentioned briefly in chapter 14, and that is herbicides. And I'll specifically be speaking on the subject of herbicide development and mode of action. So when we talk about herbicides, we're talking about weed killers, and we need to understand what a weed is, and how you define a weed will very much depend upon who you happen to be speaking to. So if you're talking to your average homeowner, and that person is trying to get weed control in his or her lawn, then basically anything that doesn't look like the grass that you're trying to grow, anything else that's in there that isn't part of that uniform, nice lawn that you're trying to produce, is considered a weed. So crabgrass, for instance, that is growing in a nicely manicured lawn, looks horrible, and that's a weed. Dandelions, which are broadleaf plants, these things are considered to be weeds, and people spend a lot of money, and probably 20% of all the herbicides that are actually in use today are being applied for that purpose, for garden control, weed control. So if you're talking to a farmer, on the other hand, a farmer's definition of a weed might be quite a bit different. And something that you might not consider to be a weedy plant, the farmer might not necessarily agree with you, and may say that, well, that's a weed. If I'm trying to grow soy and corn plants suddenly start popping up all over the place in my soy fields, then the corn is a weed. If I'm trying to grow corn and I'm getting a lot of soy popping up, then the soy is a weed. So again, it just depends on your perspective, on your point of view. Herbicide development is actually a fairly complex and time-consuming process. So it initially starts out with finding or identifying a particular compound that has herbicidal activity. I know a little bit about this because back in the 1980s, for a period of approximately 5 years, in the late 70s and early 80s, I worked as an agricultural chemist for Diamond Shamrock Corporation, which is no longer in that business. But at that time, there were a lot of people out there that were looking at screening molecules for herbicidal activity. Once you found the molecule, then you have to look at the economics of developing that molecule. So if you're going to synthesize it, how much is it going to cost you to make, and what are you going to be able to sell it for? And if you can't make a profit, then you have to really start looking elsewhere. Then if you decide that you want to develop this material, you have to look at things, you have to start doing testing on it. You have to look at toxicology. What will the effect be on, for instance, on birds, mammals, beneficial insects, if I start applying this to the plant? So we want to be killing specific things, but we don't want to be harming other things. There's also the environmental concerns. If you apply something to the soil, is it going to degrade through natural processes? Will that material degrade to compounds that are essentially benign in a short period of time? Or are we going to find out that 50 years from now, or a century from now, whatever it was that you put in the soil is still there? That's not a good thing. Once you spend a lot of money doing all these kinds of tests, you want to make sure, even before you start doing that, you want to make sure that you're going to be protected, that you're going to have exclusive use of this compound. So patents have to be filed. And so you go to the U.S. Patent and Trademark Office and you start the process of a patent application. And that can take a few years as well. You have a 20-year period. If the patent is granted to you, then you have a 20-year period in which you can exclusively develop, manufacture, and sell that product, and no one else is allowed to do that. Once you've done these things, then you have to look at developing a process to synthesize this compound. So perhaps you made this in 1 or 2 or maybe even 3 or 4 steps in the laboratory, and the way you made it in the lab is not necessarily the way you're going to make it when you're trying to go from, let's say, a few grams of the material up to perhaps thousands of kilograms, or even in some cases it could be millions of pounds of this material that you would actually be manufacturing. Once you've looked at process development and you have a good process, then you're going to be looking at things like getting registration for the compound, because you can't just make something and even though you have a patent on it, there's still a registration process, a process in which and the EPA might be involved in this, other regulatory agencies will be involved in it to actually give you not just the right to make the material, but the right to actually use the material. Once the material is registered, it can be used on certain kinds of crops, for instance, for certain types of applications. You also want to look at the formulation. So, for instance, some of the newer herbicides are active at very, very low doses. When I originally got into this area of chemistry, sometimes we had to apply as much as maybe 10 kilograms of material to an acre. And now you might be looking at some compounds where it's just a few grams per acre. You can't really apply a few grams per acre if you're trying to do that. It's such a small amount of material, it's almost going to get lost on you. So you basically formulate it so that you're going to be perhaps applying maybe a pound or so of a material that's a solid material, and only 1% or maybe even less than 1% is the active material that actually is the herbicide that you're developing. So formulation is a time-consuming but a very, very important process as well. You've got to bring this material to market, so you've got marketing activities that have to take place, and the process development, which takes place in a pilot plant, that's to make perhaps kilogram quantities of this material. But once you go into full-scale manufacturing, then there's other concerns that you're going to have. And so you have all these manufacturing types of concerns that have to be addressed as well. And this entire process, going through all these steps, could take perhaps up to a decade to complete. So it's time consuming, and it's expensive, and it's very, very difficult. So we've got different ways of classifying herbicides. Herbicides could be classified as pre-emergent, those herbicides that actually act on plants while they're still perhaps in the seed stage. They may inhibit germination, or they may, once the plant is germinated, they may inhibit root growth or shoot growth. So the plant basically is not going to get big, and it's going to wind up dying, that particular weed. That's pre-emergent, could also be applied post-emergently. Plant is now mature, you put the herbicide down. It could be contact with the plant that kills it, or it could be a kind of a systemic poison where it's taken up by the soil, it gets inside the plant, and it winds up inhibiting some particular enzyme system and kills the plant in that fashion. Herbicides that are specifically will kill monocots but don't harm dicots, the broadleaf types of plants, and we can have herbicides that will kill the broadleaves but don't harm the monocotyledonous plants. So these are all different kinds of ways of classifying things, but what I've done here is I've actually classified them, and there's probably, I think, maybe a dozen or so of these classes that I'm showing here, classifying them by their mode of action. That is, how do they work? What do they actually do? And you could also classify them as well by chemical family. It turns out that certain families, certain kinds of structures are very specific for certain kinds of activity. So this first category are called acetyl coenzyme A carboxylase inhibitors, ACCase inhibitors. And these compounds, the araloxyphenoxys and the cyclohexane diones are effective on monocotyledonous plants, that's grasses, and they kill these things by blocking the initial step in fatty acid synthesis. So all these compounds play an important role in fatty acid synthesis and metabolism, and without these essential fatty acids, the plant cannot survive. The next category, these ALS inhibitors. ALS stands for acetolactate synthase. This is an enzyme that is involved in the biosynthesis of valine, leucine, and isoleucine. These are 3 branched amino acids, and again, without these amino acids, there are particular proteins that the plant is unable to synthesize, and therefore it will die. There are 2 principal categories, chemical families: the sulfonylureas, which we'll see examples of all these things in a few moments, and the imidazolinones are the other category. And both of these compounds, classes of compounds, are involved in the same mode of action, that is the inhibition of that enzyme. So it turns out, however, that they're now finding that when people apply sulfonylureas to their crops for many, many years, it turns out that if the farmer then switches from sulfonylureas to imidazolinones, he or she may find that the weeds have now become resistant to the secondary application. So it turns out that they're now seeing a lot of these cross resistance problems that are arising from applying one of these, and then plants become resistant to the other one. And one of the ways of avoiding that is something that farmers do usually anyway, which is crop rotation. So if you've been planting corn for a period of time, you want to stop planting corn and then plant some other type of plant, and that will at least somewhat ameliorate the problems that you have with cross resistance occurring. The third category, these are compounds that have been around for a very, very long period of time. And this third category of compounds, the MTP inhibitors, are microsomal triglyceride transfer protein inhibitors, and these are actually act as mitotic poisons. So they're interfering with the process of mitosis. And specifically, it says they interfere with spindle fiber alignment, but at a more fundamental level, they're actually interfering with the production of the protein tubulin, which is what the spindle fibers are made of. So by interfering with tubulin synthesis, that these things are killing the weeds, the specific weeds that this is being applied to. These compounds have been around for a very, very long period of time, probably even before I was working in this industry. Dinitroanilins were still being used. It's one of the earliest type of herbicides that have been developed. The next category is synthetic auxins. These are the phenoxy acetic acid types of compounds, benzoic acid, carboxylic acid. We saw some of these things a few weeks ago when we talked about auxins. These inhibit cell growth, and where you've got rapidly dividing tissue, meristematic tissue, they're inhibiting the cell growth and cell division, and they kill in that particular way. Now there's some other categories that I've got here on this next slide here. These first ones we just saw, the first 4 of these here, up to synthetic auxins. Now the 5th one and 6th one are actually working or inhibiting some processes that some part of the photosynthetic process. So in this case, these photosystem 2 inhibitors, these are acting, photosystem 2 is the first, even though it's called photosystem 2, it's actually It just has to do with the way that these things were developed. But this is the initial photosystem where chlorophyll gets excited, becomes electronically excited. The electrons move to a higher energy level, and then they fall back down to the ground level through a series of steps. And as they do that, there are a variety of other molecules that are involved, things that are, the energy is being captured and it's being used to synthesize other compounds. But inhibiting photosystem 2 or photosystem 1 winds up killing weeds. And the 5-enol pyruvyl shikimate 3-phosphate synthase inhibitors is another category that we're going to look at. Glutamine synthesis inhibitors. These are a class of compounds that prevent glutamic acid from being converted into glutamine, which is another essential amino acid. But more importantly, one of the things that happens when this occurs is that high levels of ammonia can build up in the plant, and that becomes toxic to the plant if this enzyme is the glutamine synthase, which is the enzyme, is actually inhibited. And these last, I believe it's just these last four, deoxyxylulose 5-phosphate reductoisomerase inhibitors, another category that I'll talk a little bit about, 4-hydroxyphenylpyruvate dioxygenase inhibitors, again, specific enzymes, and we'll talk a little bit about what they do. And protoporphyrinogen oxidase inhibitors. These are the protoporphyrinogen oxidase is actually involved in the synthesis of chlorophyll. So again, what's happening when you apply these things, one of the things that you notice when you apply these to plants is that the plants bleach, they become white, and they're unable to carry out photosynthesis, and then they die. And very long fatty acid synthesis inhibitors, the long chain fatty acid synthesis inhibitors, again, the long chain fatty acids are required for the synthesis of essential oils, kind of the fats of the plants, and again, without these things, the plants cannot survive. So these are different ways in which you can kill a weed. And so I want to look at some of the specific categories and a little bit more at the molecular level of what these things do. These pyridiniums, and what they do is they capture electrons from photosystem 1 and they form free radicals. And these free radicals are highly reactive intermediates that contain, that actually contain a single unpaired electron. It makes them very, very reactive, and they wind up destroying cell membranes. And so they're killing cells in that way, and then the plant ultimately dies. In the photosystem 2 inhibitors, we have more classes of compounds. Triazines, which have been around for a very long period of time, nitriles, and there's one specific nitrile that I'm going to talk about a little bit in more detail, a compound called diclobinil, or casseron, which is something that I worked on myself for quite a long period of time. The benzothiodiazinones and the ureas. And all of these things have similar modes of action. They block electron flow in the process. These are some of these intermediate electron capture molecules, Q sub A, Q sub B, and this stops carbon dioxide fixation, which means that you're not going to be able to produce ATP or NADP, PH2, the reduced form of NADP, and again, that's going to wind up killing the plants. Another category chemical family is the diphenyl ethers, and they inhibit chlorophyll biosynthesis. Compounds that are involved, or all the compounds that are shown here, the diphenyl ethers, the N-phenyl thalamides, and the aryl triazinones. And these are compounds, the names of the compounds that a farmer would actually buy: Blazer, Reflex, Cobra, Resource, Valor, Spartan, AIM, and Cadet. Those are the protoporphyrinogene oxidase inhibitors. And other enzyme inhibitors, the EPSP synthase inhibitors that are shown here. This is the enol pyruvyl shikimate 3-phosphate synthase inhibitors, and they inhibit the this synthase that enables production of aromatic amino acids. So this first category of compounds prevents the synthesis of things like tyrosine, phenylalanine, and tryptophan, 3 essential amino acids. Again, it means that the plant can't make specific proteins. So when that and it turns out that there are a number of compounds that are actually related or derivatives of the naturally occurring amino acid glycine. So these glycine derivatives act as inhibitors of amino acid synthesis. Those specific amino acids, tyrosine, phenylalanine, and tryptophan. The glutamine synthase inhibitors, a category of compounds called phosphinic acids, and these are actually also glycine derivatives. They stop conversion of glutamic acid to glutamine, and I mentioned this before. This leads to a lethal concentration of ammonia developing in the plant cells. This next category, these DOXP synthase inhibitors, what they do is they interfere with isoprenoid biosynthesis. The isoprenoids are compounds that will ultimately be used for the synthesis, for instance, of phytosterols, things like beta-cytosterol. These are the plant sterols, and again, these are necessary for the plants to be able to grow and develop normally. The isoaxolidinones are a chemical class that have that particular activity. And then we have the 4-hydroxyphenylpyruvate dioxygenase inhibitors. They prevent tyrosine catabolism. So the amino acid tyrosine needs to be broken down, and in the process of breaking it down, it's used biologically used to synthesize carotenoids. And these compounds, the pyrazoles and these triketones, have that activity of inhibiting that dioxygenase, and therefore the carotenoids cannot be synthesized. And one of the things that the carotenoids do, things like beta-carotene, protect chlorophyll from photo destruction. So again, when you put these things down, it's going to cause bleaching. The next category are fatty acid synthase inhibitors, and these are things like the chloroacetamides and oxyacetamides, and they inhibit very long fatty acid synthesis in the again in the growing meristematic shoot tissue. And here we're actually seeing some of the chemistry. So these are selected herbicides by class and by mode of action. And a couple, I put down the trade name here, the chemical structure. These are 2 of the ACCase inhibitors. Tetraloxydim is one compound which actually probably be categorized as a cyclohexenone type of compound. This is cyclohexene right here. This is a cyclohexenone. So that's how we would categorize that compound. This is a phenoxy type of structure. Here's the phenyl and the oxygen over here, so that's a phenoxy. And these things are, as I said, are ACCase inhibitors. And this is a HPP dioxygenase inhibitor, sulfotrione. This is a triketone. So you saw on a previous slide we talked about a triketone. So these C double bond O groups here are ketonic groups. Now, I have to tell you that back in the 1970s and the 1980s, the way compounds got developed back then, and when I was working at Diamond Shamrock, we had 2 groups. One group was a pharmaceutical group, and the other one was an agricultural chemical group. And I happened to be in the agricultural chemical group. But it turned out that every time we made a new molecule, and you might have had whatever the chemist's reason was for making that molecule, perhaps it looked structurally similar to something else that was already on the marketplace. So you thought, well, for instance, if this was a molecule that worked, I might want to try changing some of the things here. Maybe I could swap for the oxygen. I could put a sulfur here instead of the oxygen. Or maybe I could change the C double bond O and have an SO2 here. There's other kinds of things that I could do to the molecule. Like I have an SO2 over here. There's other things that I could do to the molecule. And I might make that molecule and see whether or not it had that particular kind of biological activity. And that's a lot of what we did back then. But it's a very, very inefficient way to develop a new kind of compound. And the other problem with doing that, let's say sulfotrione is a very, very good compound. It's owned by one of my competitors, and I'd like to be able to make sulfotrione. Well, I can't make it because it's patented. And probably I can't even make the compound that has the sulfur over here, because when they patented it, if they did it correctly, they claimed not only this compound, but a whole bunch of similar compounds. As a matter of fact, they looked at other, what we call analogs, things that are kind of structurally closely related to this, and they specifically claimed those compounds as well. Even if they didn't do anything, they probably claimed those compounds. That's what we did as well when we developed new herbicides. We would claim everything under the sun. So basically, that's what you did. And it was kind of a hit or miss type of approach. You made a molecule, you tested it. We would test it both in the pharmaceutical area and also test it in the agricultural chemical area for a wide variety of screens. We would take this compound and we would screen it as a herbicide. We would also screen it as a fungicide. We would screen it as an insecticide. We would screen it as a nematicide, something that kills nematodes, these roundworms that get into the soil. So we had all these different screens, and then it would be screened for cardiovascular activity and central nervous system activity and a variety of other things in our pharmaceutical group. So again, not the world's greatest way of actually coming up with a new compound. A better way of doing that is at specific enzymes, and perhaps once you understand what the mode of action of a chemical agent is, usually what that's doing is the structure of this compound is such that it is going to fit into the active site of some particular enzyme, and it's going to bind to that enzyme. And it's going to do it, there's going to be two principal things that we're going to look at. One of them is the stereochemistry. That is, what does this molecule look like in 3 dimensions? What is its shape? How will it actually fit into that 3-dimensional hole or area within the enzyme where it has to bind? Where normally whatever the normal substrate is that would normally bind in there is being blocked by this compound. So we want it to be what we call isosteric, which means to have about the same stereochemistry as the naturally occurring substrate that would fit into that active site. And we'd also like it to be isoelectronic. So that means that we want to have, there's going to be certain areas of the active site that will be polar and other areas that will be nonpolar, and using the principle of like attracts like. So this is a polar portion of this molecule that would perhaps bind to the polar portion of the active site, and this part over here is nonpolar, so that might bind to another portion of the active site. So we would look at things like that, and we would try to design a molecule that would fit into the active site and do specifically what it was that we were trying to do. And you still had to make these things, and perhaps I could synthesize 2 or 3 compounds a day back then, and it might take up to a week to actually test those things. With the new synthetic techniques and the new screening techniques, that is, how do we actually check these things for those particular kinds of activities? A lot of these things can actually be put on computer chips right now. They can be synthesized and tested, and it's possible to synthesize hundreds or even thousands of these things in a day and screen them very, very rapidly. Screen as many of these compounds as you actually make. Here's a photosystem 1 inhibitor called paraquat. Ramoxone is the generic name for this compound, and it is one of these bi or di pyridylium types of compounds. You'll notice it's a charged molecule here, has a positive charge on both of these nitrogen atoms. This is pyridine, and this is pyridine here. So the compound is referred to as a 4,4' dipyridyl, or dipyridylium, because it's this ion, and this is a dichloride. And this compound, which is very, very easy to make, it's fairly toxic to human beings. You would not, I don't believe you would actually ever use this around crops. The Mexican federal government was actually spraying this when they would come across marijuana fields. They would spray this stuff on the fields. Even if it didn't kill the marijuana, it made anyone that would smoke that marijuana would get very sick. And I think that the reason for doing that was maybe they didn't even do that, but they spread that rumor around that they were doing it so people would stop using the marijuana. Obviously, it didn't work. Photosystem 2 inhibitors, and one of the compounds that I'm showing on the slide, there are 3 different categories here. This is a betazine or basagran compound that you see over here. Atrazine is a triazine. This again is a compound that's been around for a very, very long period of time, and it's still being used quite a bit. It's used, I think it's frequently used as a corn herbicide. Linuron is an example of the urea. A urea is a compound that has a carbonyl, C double bond O. Bonded to that are 2 amino groups. Urea itself would be NH2 and NH2. These are substituted ureas. And another compound here, which is called diclobanyl, casseron. This is a compound, this 2,6-dichlorobenzonitrile. Diclobanyl is a compound that I myself became very, very interested in because of some work that I had actually done on my PhD dissertation involving this molecule that you see over here. And I knew that this molecule, we weren't interested in this. We were interested at that time in this molecule, which is a benzene sulfenic acid. These are very unstable molecules, and I spent years studying the chemistry, what happens to these molecules as they decompose. There's only maybe perhaps a half a dozen, or maybe at most 10 or 12 of these things, I would say, that are actually known to exist that are stable. But almost all the other ones are unstable. Nevertheless, they have important biological implications. So I was looking at some of these things, and one of the things that I found out where we synthesized a number of compounds like this, and found out that if we heated them up, they would undergo this particular reaction that eliminates a sulfenic acid, and the other product of the reaction is a nitrile. So I wasn't necessarily looking at the 2,6-dichloro. That in my thesis work, I don't think I even made this compound. But when I got into the actual herbicide synthesis business, and we were interested in molecules like diclobanyl, and I had the idea that perhaps since diclobanyl was patented and we couldn't use diclobanyl, perhaps we could use this as something that is sometimes referred to as a pro-herbicide. That is, let's say if we put this down into the soil and over a period of time this reaction would occur spontaneously, it would actually be kind of a slow release of diclobanyl into the soil, and perhaps that would kill plants. Well, when we started to look at a whole bunch of analogs, we put a lot of these, R just refers to different groups, and since it's not, I'm not showing you where the group is, it's just a variety of groups that are attached at different positions on the ring. And so the idea was to look at all these different molecules. And when we did that, we found, I published a couple papers on this, one by myself and one in collaboration with Dr. Anton Hopfinger, who was a professor at Case Western University. And Tony was looking at modeling these compounds, their conformation. That is, what does this molecule look like in 3 dimensions? And it turns out that we found that there was a correlation between those forms of the molecule where the most stable form was planar. That is to say that this ring and this, all these things kind of were flat in a plane. If you got, in some cases where you actually got this part of the ring to twist. So we looked at twisting the molecule around this bond, and we found that the most stable conformation was the one where this was planar. But what we found is that as we made other analogs of this, any time that the molecule was twisted out of its planar conformation, it lost its biological activity. But what was more interesting was that these compounds did not look like diclobinol. They were much more selective. Diclobinol basically kills everything. It doesn't have very good selectivity at all. So this is another one of these compounds that you would use in right-of-way situations where you basically have a piece of land and you just don't want weeds growing on it, but you're not trying to grow crops or anything else on it. Maybe you don't want these things coming up, you don't want weeds coming up in your sidewalk or something. So you could put diclobinol down. There's a lot of other things, newer compounds that work better than that now. But anyway, so looking at this particular molecule, we found that these molecules were much more selective, and so it was evident that this molecule by itself, this whole molecule, had some kind of biological activity that was not due to its decomposition to diclobinol. So it wasn't just forming diclobinol, because if it had been, then it would have had the same kind of selectivity that diclobinol did, but it didn't. And it was in some cases, some of the molecules we made were more active than diclobinol. And there was one specific molecule where the hydrogen, I replaced the hydrogen with a methyl group, with a CH3 group, and that compound had an entirely different mode of activity than any of the other compounds that had hydrogens in it. It had different, entirely different selectivity. So it was much different than either the compounds that were shown here with the hydrogen, and it was also a lot different than diclobinol. Most of these things were broadleaf herbicides. Diclobinol was active against broadleaves, and this compound with the methyl group here was a grass herbicide, but it had some broadleaf activity as well. So that was kind of interesting. It never got developed, by the way, because shortly after this, Diamond Shamrock sold its agricultural chemical division, and I think some of these molecules that I worked on actually got lost in the shuffle. Here are some ALS inhibitors. We talked about these before. These are the acetolactate synthase inhibitors, the sulfonylureas. Again, this is the urea. Remember, there's 2 nitrogens and a C double bond O, that makes it a urea. And this SO2 group is the sulfonyl group. Anything that had that functionality, SO2, NH, CO, NH, those kinds of compounds were, and are, I should say, ALS inhibitors. Here's a compound called Scepter. Imazaquin, it's an imidazolinone. This is called, this 5-membered ring with 2 nitrogens is an imidazole, and this makes it an one. Imidazolinone. Another one called imazapyc, and imazapyc differs in the fact that it has a pyridine ring attached to this. The rest of the molecule is the same, and here this has a quinoline ring attached. And the last one is a pyrimidine. So this is a 6-membered ring with nitrogens at the 1 and 3 position, pyrimidine. And remember from our discussion of DNA, these are things like uracil and thymine and cytosine. These are the pyrimidines. So these things are all ALS inhibitors. You've got the dinitroanilines. I talked about these before. Trifluralin has been around for a long period of time, and this is a methyl aniline, the methyl group being the This is actually a trifluoromethyl group, but they're referred to as methylamyl. When it's methyl, it will be a CH3. This is a CF3, synthetic auxins. This again, these are some compounds that I also had some involvement with. Dacthal, the compound that you see down here, is actually the DAC part of this name comes from Diamond Alkali Company, which was the precursor to Diamond Shamrock. So this was developed by Diamond Shamrock chemists, and this particular compound is a terphthalic acid derivative. That's this benzene with 2 CO2H's on here, and it's a tetrachloroterphthalic acid compound. When I used to go down to our plant on the Gulf Coast of Texas, and I would come home after being at the plant for about a day. Sometimes I would have to go down there for business meetings, or sometimes to actually inspect certain aspects of the production of compounds like this at the plant. And when I would come home, my wife would basically make me take off my clothes in the garage before I would come into the house, because I would smell so bad from these compounds, which were fairly volatile, and they would just get into your clothing. And my wife would, before she could even send my suit to the cleaners, would have to let it sit in the sun for maybe about a week before the odor dissipated enough that she could take it over to the cleaner without the cleaner saying, "I'm not taking this in, it stinks." So that was Dacthal. And dicamba is another benzoic acid derivative, Banvel, a compound that is still a pretty big seller. Stinger clopyrrolide is a pyridine carboxylic acid, so it's like a benzoic acid. It's a little different because it's got the nitrogen in the ring, but it's got the CO2H again. And here is 2,4-D, 2,4-dichlorophenoxyacetic acid, and there was also a compound called 2,4,5-T, which had a chlorine over here. 2,4-D is still used. 2,4,5-T is not used anymore because of problems that are associated with the fact that that particular compound, in its manufacture, there were always small amounts of a compound called 2,3,7,8 tetrachlorodibenzoedioxin that were formed. And for many, many years, the EPA insisted that this was one of the most poisonous substances on the face of the earth, and they came up with regulations that said you couldn't have any of this in your product at all if it was detectable, and you could detect it down to femtomolar quantities, like something like maybe a trillionth of a mole or something like that. You'd still be able to detect it. So it was impossible to get rid of it. And it turns out that the EPA has lightened up a little bit on this because it's now known to not be nearly as bad. It's very, very toxic if you're a guinea pig. This kills guinea pigs at very low concentrations. It doesn't seem to do anything to human beings, and not this compound, but I'm saying the dioxin. It doesn't seem to do anything to human beings other than cause a very severe skin condition, which actually clears up after a period of time. It's called chloracne, and that's the only real human health concern from these dioxins. But nevertheless, I also spent a large portion of my career developing processes, a chemical process that would actually destroy dioxins, and that was for an entirely different chemical company. But I've got patents for getting rid of dioxins. Once they wind up in the soil, we can treat the soil and destroy the dioxins. But I don't think dioxins are what we call a political poison, and I don't think anybody's terribly worried about them anymore, except maybe some of the foaming at the mouth environmental groups. These are two compounds that are somewhat related in terms of structure, but their mode of action is entirely different. So the first one is a compound that we talked about already in a previous lecture, and that is Roundup. We mentioned that Monsanto had developed a compound called glyphosate, and it's a big seller. And one of the things that they wanted to be able to do is to be able to apply it, since it was something that wasn't very selective and pretty much kills everything that it comes in contact with. They were trying to develop specific seeds. In this case, corn seeds were the first one. And now we know that there are other so-called Roundup ready crops that have been developed, but things that were resistant to Roundup, whereas the other weeds would not have that resistance, and the weeds would be killed by this material. So this particular one is a 5-enol pyruvyl shikimate 3-phosphate synthase inhibitor, an EPSP synthase inhibitor, and that inhibits the production of tyrosine, phenylalanine, and tryptophan, the aromatic amino acids. So it kills everything. And it's a derivative of the amino acid glycine, which is amino acetic acid. So here you have the carboxylic acid group, the COOH. This would be a C, if this were a CH2 and this were an NH2, that would be glycine. So we've got a CH2, and then this is called a phosphonic acid group. So this methyl phosphonic acid group that's attached to this glycine molecule makes this glyphosate, the EPSP synthase inhibitor. Here's another compound that's called glufosinate, or that's the generic name, and I'm sorry, I think that's a trade name, and phosphenothricin is the generic name for this. And this is the glutamine synthase inhibitor that I alluded to before. It's also still a derivative of glycine, but here, rather than this would be the glycine molecule. This would be a hydrogen here. So here, the attachment of the other group was on the nitrogen of the amino group. Here it's on this carbon, this alpha carbon of the glycine molecule. And again, what we've got here is not a phosphonic acid, but a phosphinic acid. There's one fewer OH group here, and there's a methyl group here, and then there's an ethyl group here that is actually attached to this position here, and this is glufosinate. So it's a somewhat similar kind of compound, but again, it's structurally different enough that it has an entirely different mode of activity. So this is the glutamine synthase inhibitor, the one that prevents the conversion of glutamic acid to glutamine. So glutamic acid and ammonia in the presence of that enzyme would give us glutamine, but glutamine cannot be synthesized, and that again causes toxic levels of ammonia to build up inside the plant. And here's acetochlor, and I worked with these molecules as well, a bunch of analogs of this that had, where we took the this group off here and we put on another CO and something else out here, various other functional groups, but we made a bunch of those as well. And they had some herbicide activity, but they weren't great. Acetochlor is a pretty good herbicide. It's a pretty active herbicide. And again, it's a compound that quite a bit of that winds up being sold as well. Here is a compound called Command. This is a oxazolone. That's the name of the ring system. This oxygen, nitrogen. So this would be called oxazolinone is the name of that ring system. So they have fairly complex names, but this is sold under the name of Command, or clomazone is the generic name for this particular compound. And this is the one that interferes with isoprenoid biosynthesis. So you put this down, and then you can no longer make phytosteroids and other essential plant components. And these are some of the 4-hydroxyphenylpyruvate dioxygenase inhibitors, which prevent tyrosine catabolism, and therefore prevent carotenoid synthesis. And that leads to the destruction of chlorophyll. Two different categories. One of these is an ice. This again is has an iso-oxazole ring on it, and this over here is a pyrazole ring. So these are the two rings that are on this molecule. But this one here is definitely considered to be a triketone. So this is mesotrione, trione indicating that there are 3 of these carbonyl groups, and this is called callistic. And here we have a class of PPO inhibitors, clomethoxanil, which is a phenoxy compound, flumioxazin or Valor. This compound is a phthalimid, this is a sulfonamide. It's also a triazole, and this is called Spartan. So these are all PPO inhibitors. So I just wanted to show you these, that last one being a porphyrinogen oxygenase inhibitor. That was the PPO that inhibited chlorophyll biosynthesis. So this is just to show you what goes into having to develop a herbicide and the way that they actually prevent plant growth, how they inhibit weeds specifically, and we'll be talking in, I believe, next week on insecticides, where 80% of the development of herbicides goes to the agricultural chemical industry, and worldwide that's really hundreds of millions of pounds of this stuff that's actually being applied. So, you know, it's pretty big business. It's probably, if I had to guess, I think maybe the entire pesticide production is maybe $40 billion, probably something on the order of maybe up to maybe 15 billion of that would actually be involved with herbicides. So there's a lot of financial incentive for making these kinds of compounds. And we're going to be looking at insecticides next week. So again, thanks for your attention, and we'll have the usual 25 question exam for this.